By following this protocol, the user will be able to produce AAV vectors of high purity and yield, suitable for in vitro or in vivo applications. This protocol can be used to produce and purify a range of AAV serotypes with different transgene configurations. Combining these two elements can be useful to obtain cell type and tissue-specific expression. Production of some serotypes can give to lower yield. Ease of production varies across serotypes and should be evaluated on an individual basis. Demonstrating these procedures will be Shelly Fripon, a technician from our laboratory.
After growing the HEX cells, mix 360 micrograms of helper plasmid, 180 micrograms of plasmid encoding the vector capsid, and 180 micrograms of plasmid containing the transgene in 18 milliliters of 150 millimolar sodium chloride to prepare the DNA mix. Distribute this mix over 3 50 milliliter conical tubes. In a new conical tube, mix 840 micrograms of PEI in 6 milliliters of 150 millimolar sodium chloride to prepare the PEI mix for 6 cell culture dishes. Then, add 6 milliliters of the PEI mix drop by drop to one of the conical tubes containing the DNA mix. Incubate at room temperature for 20 minutes. Next, remove 6 cell culture dishes from the incubator. In a laminar flow hood, completely aspirate the medium from each dish. Rinse the dishes with 5 milliliters of pre-warmed DPBS to remove traces of the medium. Gently tilt each dish to ensure that